be caused by pollution, loss of habitat, or deforestation. Could be caused by human impact, loss of diversity, poaching, global warming, pollution, release of CO2 in the air, loss of the ozone layer, same thing. Introduced species, human activity, kind of similar thing. Also, industrialization would have to do with human activity. Just basic human activity and the increase of human population could have something to do with it. So let's just go over some basic terms here. The energy pyramid shows how energy is used up with each step on the food chain. Complex food web is pretty much what eats what, as I'm sure you know. Ecological succession is a series of steps in which new communities replace current ones, until a stable climax community is established where everyone pretty much has what they want and everything is good. So here is now what you did during the state labs, a little bit of a refresher for you. These are the 4 state labs that will be in the final part of your regions. So, part A1 in the Making Connections, aka the Clothespin Lab. What you did is you measured how exercise affected pulse rate. Exercise increased pulse rate, as you learned. What you did is you squeezed the pulse. Part A2 is what you did is you squeezed a clothespin for a minute and then squeezed it again for another minute. What you learned is if you squeezed more the second round, it may have been because your fingers were warmed up from the increased circulation. If you squeezed less the second round, it could be because your finger muscles could have been fatigued. What you did, you designed an experiment to test how exercise affects squeezing a clothespin. That was in part B. What you learned is how to kind of design an experiment, reformulate, and create future experiments in the end. So relationships and biodiversity. This is Botanic Curist Lab. What you did is you compared 4 species of plants based on structural and molecular traits. What you learned, the species that are related share similar traits. Different techniques such as gel electrophoresis and paper chromatography can be used to determine relationships between organisms. Endangered species should be protected because they offer many benefits to humans. What you did was you played different finch species competing for food in the Beaks of Finches labs. What you learned, different environmental conditions or food favored different species of finch, allowing them to survive and reproduce. But some others were not so lucky. Diffusion through a membrane. What you did there is the diffusion through a membrane lab is what you did is you made a model cell using dialysis tubing. You put glucose and starch inside your cell. You put the starch indicator, iodine, outside your cell. What you saw is the inside of the cell turned black because the iodine diffused into the cell. Because outside of the cell was not black, you know the starch did not diffuse through the membrane. You used blue glucose indicator or a Benedict solution to see that the glucose did diffuse through the membrane. What we learned is the small molecules can diffuse, large molecules cannot. You can use indicators to identify the presence of specific substances. And what you did in part B of that lab was you looked at red onion cells on the microscope. You added salt water to the onion cells, added distilled pure water to the onion cells, and you saw salt water cause the onion cells to shrivel, and distilled water cause the cells to swell back to normal. So everything kind of rebalanced that effect there. And all right, that has been your bio podcast, Everything Living Environment, in just 49 minutes.